Everybody, we're going to jump into the next set of notes, get started right away. Let me shrink myself down here real quick. Oops. There we go. All right, so we're still talking about evidence collection, focused on biological evidence this week. So the learning objectives are pretty much the same as they have been. We're just focusing on a different type of evidence now. Your assignment for this week is going to be a short 500-word essay on how environmental factors could affect DNA evidence. So your focus could be any number of various things. It could be through contamination, different things that could contaminate any kind of DNA evidence, or through the natural degradation of the DNA itself due to environmental factors such as temperature or moisture or those type of things. So the 500 words is just the general guideline. If you're a little less, if you're around 450 or something, or you go a little bit over 550, you're okay. So just try to hit that 500 word range. All right, so let's jump into this. A quick review of a history lesson. I'm just going to kind of briefly go over this. In the early 1800s, that's when serology kind of made its start, right? The first attempts were made to identify different stains, reddish brown stains on clothing as blood. Most of these focused around microscopy or various chemical tests for the presence of hemoglobin. And these were some of the first presumptive tests, and we still do a lot of these simple presumptive tests even today, like the Kastle-Meyer test, which is one of the ones we would use in the lab for our purposes, which is using phenolphthalein and hydrogen peroxide to see a color change in the phenolphthalein if there's hemoglobin present. And then a little later, we started coming up with the first confirmatory tests that were starting to be developed. And then in the early 1900s, 1901, we really started getting into protein analysis. And this protein analysis, you're going to see, goes on for about 60 or so years, 70 years. And right around the same time we get towards the end of that is when DNA starts to come into its heyday, and it's still there now. So in 1901, the first methods were used to distinguish animal and human blood by using the various proteins that are in both animal and human blood samples. And then we also, around the same time, started coming up with, from Landsteiner, the ABO blood types. So prior to this, if you had type A blood and they had to do a transfusion, and it's kind of like rolling the dice. Sometimes the transfusion could actually kill you, and they're trying to save your life. But that was because they didn't realize that there was different blood types, right? So now we know about the A, B, O, and AB blood types that are out there. And then short time after that, right around 1915, we're still doing, developing proteins, right? And the next big one was the Rh factor. So you know how we talked about blood types. Well, you have blood type A, but you can be A positive or A negative, and that's the Rh factor. It's called rhesus factor. It was discovered in rhesus monkeys, and that kind of helped even further with blood typing, because now we could go A positive, A negative. And then we have all these other tests that were focused around various proteins, like phosphoglutamase. You had all these different types of proteins that you can find in blood, and again, this is all kind of focused around these 50s and 60s. But right around this same time, right around 1958, we started having Watson and Crick developing their model of DNA, and we started kind of moving away from proteins and towards genetics, DNA. And that's where we end up in the 80s. Sir Alex Jeffreys did the seminal work in DNA fingerprinting, and it was first published in scientific literature. In a very short time after that, I think this was a case in England with Colin Pitchfork, was the first time that DNA profiling was applied to a forensic case. And then after that, right around the same time, same year, polymerase chain reaction, PCR technologies were invented and started to be used to replicate DNA. And that allowed us to use very small samples that can be found at a crime scene to actually replicate it and use it to test various suspects and try to do a comparison. And it goes on after that. I didn't go into too much detail, but shortly after we started being able to use mitochondrial DNA and all that. So let's move away from that brief, real quick review of history of serology and kind of start talking about how we handle this stuff at a crime scene. So blood and other biological fluids may be recovered as evidence in violent crimes. Typically, you're going to have these bodily fluids and tissues and blood in crimes that are going to be, murders or rapes or some kind of assaults or something like that. So the evidence is commonly found as blood from a victim on the clothing of an assailant, or as semen from an assailant recovered from a rape victim, or perhaps a series of blood spatters that could be left at a crime scene. And we're going to handle blood spatter a little bit later. Originally, I was going to kind of cover it right at the same time, but hopefully with us moving to on campus, it might lend itself a little bit better if we wait and do some of these labs in a laboratory setting. So we'll see what that brings. Less obvious forms of evidence, however, can include body fluids such as saliva, urine, vomit, amniotic fluid, body parts, human remains, tissues associated with like mass disasters and homicides, or injuries inflicted by vehicular accidents or hit and runs. So any of those are going to have obviously blood or various body fluids in human tissues. So although such examples contain markers of individuality, the individualizing test may be hampered by sample degradation. And this is where your research is going to come in. The occurrence of mixed fluids, poor sample integrity, the challenges of analyzing samples mixed with chemicals, textiles, dirt, all become problematic. You can also have degradation through environmental factors such as high temperatures or moisture or improper packaging, and we're going to go into that a little bit as we go through the notes. It's the responsibility of a forensic serologist to kind of look at the blood. Usually they're working in a lab. However, again, sometimes these serologists will come out and do blood stain and blood spatter examination, but most of the time they spend their time in the lab. So it's up to the CSI to kind of properly package these materials and get them into the lab so that the forensic serologist can actually analyze their findings. So let's do a quick review, and you guys are getting pretty good at this based off of what I've seen in some of your assignments so far. Just as a quick review of how we process the scene. Again, securing the scene, obviously you're going to photograph and document the scene. You're going to make sketches. You're going to record and do all that. You're going to start, analyzing the physical evidence. And really, if you remember, we start with the very transient evidence, like trace evidence, and then we move probably directly to sampling for biological evidence next. And the reason you want to sample for biological evidence next is because you don't want to do anything like fingerprinting where you're going to get fingerprint powder mixed in with the sample. So you're going to want to take samples of anything that might have DNA before you move into the other collection avenues and start dusting for fingerprints and that type of stuff. So how are human remains investigated? Usually if you have a violent crime, you're going to have a body, or at the very least a victim, but you could have a body, a homicide in homicide situations. So when we're investigating a homicide, the forensic investigator has the responsibility to help establish the nature of the death, whether it's accidental, homicide, or suicide. And they have to do this while preserving any evidence that's present at the scene. Moving the body, clothing, or any other items covering a corpse may result in the loss of evidence. So the very first thing you're going to do is you're going to document. You're going to photograph this body. You're going to make sure that you kind of sketch it and you make notations of the state of the body. Is the body fully clothed? Is it undressed? Is it, what kind of wounds are present? What position are they? Are they face up? Are they face down? Does it look like the body was moved after the injuries? Is there evidence that maybe the blood was smeared in a way that looks like the body was dragged? You have to document all of this. Was there anything in the person's hand? The weapon that might be on the floor, and I'm kind of referring to the picture now that's here. What was there a weapon present? Where was the weapon? How was it positioned in relation to the body and all of that? So, and I just kind of covered this, but this is kind of a list that I just talked about when I was looking at that picture. So, the position of the body, the description and state of clothing, location and intensity, direction of flow of any staining, the type of stain, any visible damage to the clothing or body, description of blood staining, appearance of evidence, removal, was something removed from the scene, estimated amounts of blood. All of that has to be documented, and it has to be documented with sketching, photographs, and actual written notes. Okay. All right. So let's look at some safety concerns, and then we'll talk about packaging at the end. When processing a scene, an investigator may face a number of potentially hazardous situations. The most common hazard faced by forensic serologists is going to be exposure to potentially infectious human um, blood and body fluids. Right? There's several avenues of exposure. You could inhale this stuff. It could be through skin contact, ingestion, accidental injection. If you're dealing with hypodermic needles from you know, uh, scene where there were drugs involved and somebody accidentally pokes themselves, they could get contaminated that way. And there's various ways, right? The main thing they have to look out for are things like um, bloodborne pathogens, HIV, um, any type of hepatitis, and those type of things are a concern. Now, the good news for us, if we deal with any of this, is all of our blood is synthetic. It's not real blood. So we don't have to worry too much about this, at least when we're practicing at this level. But if you ever do this job in the future and you're working a crime scene, you got to be aware of these. So the main thing is PPE. Make sure you're using the proper protection for all of these types of scenes. So a forensic investigator routinely comes into contact with body fluids, tissues, and biological specimens that may be contaminated with these bloodborne pathogens. Pathogens pass from biohazardous materials and uh, waste can cause life-threatening, debilitating diseases such as HIV, AIDS, and hepatitis. So you have to be aware of these. And the main way that we protect is using these universal precautions, right? Make sure you have barrier protection. Disposable gloves, coveralls, shoe covers must be worn when handling potentially infectious materials. Appropriate eye and face protection, whether it's a full face shield or safety glasses or something like that, that'll help alleviate any kind of splatters that could get into your eyes. Proper disposal of sharps in containers labeled with biohazard warning. This one seems kind of obvious, but we'll mention it anyway. No eating or drinking. You shouldn't be investigating a crime scene while you're walking around with a ham sandwich. And if necessary, respirators, especially if we think there could be any kind of uh, airborne virus or chemicals that might be present. So, how is blood searched for at a crime scene? Obviously, blood's not going to be obvious all the time. You would think, oh, you just go look for the red fluid that's on the ground or whatever. But keep in mind that a lot of times you're going to have crimes where some of this evidence tries to get covered up. So biological evidence is most informative in cases when a suspect and victim come into close contact. If I flip back a couple slides to this, the lady in the picture that was stabbed, um, if she died from a stab wound from that knife, then the person that was holding the knife probably had to be close enough to her to where he was going to have blood on his hands, maybe on his clothing, or anywhere else. And obviously, that can even be transported unintentionally by the assailant to door handles or car steering wheels or even to their place of residence. So when we're looking at this, obviously these stabbings or beatings, the assailant and the victim are going to be in close proximity to each other, and you can have a transfer of blood between the two. And it can go the other way. If the victim is defending themselves, then maybe the assailant gets injured and it could get a cut, and the blood could be transferred from the assailant to the victim as well. So we have to be aware of that. When processing a scene, the most fragile evidence obviously collected first, and then the more obvious evidentiary items are identified next. And one of the main ones we would do before any kind of fingerprinting would be looking for taking samples from biological evidence for DNA. So how are these visualized? Because obviously in some scenarios, as I mentioned before, it's not going to be unusual for a perpetrator to try to conceal or hide evidence by cleaning up blood contaminated areas. However, blood's actually really difficult to remove from the scene completely. Since blood's a fluid, it can flow between floorboards or under baseboards. It can get into carpeting, and maybe the surface of the carpeting looks clean, but it gets into the padding underneath, and we can visualize it using some special techniques. In such cases, it may be more useful to conduct searches by using high-intensity light sources that can aid in the visualization of the bloodstains or other bodily fluids, thereby minimizing loss of the evidence at the scene. And I'm going to show you one really cool one with luminol in a little bit, but let's focus on the light sources first. One thing we ordered for next year are ALSs, and I can't remember if I ordered one or two. They are really expensive. They're about $500 or $600 for a little tiny case that basically has various ALSs of various wavelengths. And we ordered one for next year. We don't have one this year. We have some low level, black lights and fluorescent lights, but nothing like the actual ALSs. Because ALS techniques can vary for a variety of types of evidence, and remember, we can use this for trace evidence, we can use it for, blood stains, we can use it for biological stains, we can use it for a whole lot of different techniques. It has to be considered, one of the better options for trying to detect hidden evidence or hard to find evidence. We consider cost, color of wavelength, all that has to be kind of taken into consideration. The light intensity, portability when we're using this, voltage requirements, safety. One of the safety things is you always have to wear a filter with these things, even though you're not shining them directly into your eyes. Never do that. You would be shining them on a surface that are so intense that even the reflected light coming back to your eyes can actually hurt your eyes. So you always have to wear filters, and usually the filters help with the visualization as well. You have orange filters, you have clear filters, you have all different filters, and they're in the form of like these little safety glasses that you would put on. All right, so talking about the light source, to enhance a stain in contrast to its background, the light must shine on the stain in a way with little or no reflection, especially if we want to get pictures, right? We don't want to get any terrible reflections coming off of whatever the object might be back to our camera. The wavelength of light used must be a wavelength opposite to the color of the stain. The detection of the blood in the obscure area can often be carried out with a high-intensity light, enabling samples to be located, obtained, and then taken to the laboratory for further testing and confirmation. Here is a really cool substance. It's got some drawbacks, and we'll talk about those, but luminol. Luminol is really cool, and I actually in one of the scene descriptions that we were talking about for one of your assignments, I was hoping to kind of lead you guys in this direction. Some of you guys got it, and many of you kind of missed it or thought it was something else. But in some crime scenarios, stain visualization enhancing chemicals might be used to locate and identify areas where suspected blood staining occurred. The most popular chemical to use is something called luminol. This stuff's really cool. In dark, when you spray this on blood, it actually glows in the dark. Even if the stain's very diluted, I mean, you might not see it. And I'm going to show you a picture on the next page that shows the same picture, one normal and then one with the light off after it's sprayed with luminol, and you'll see a big distinction between the two. When luminol is applied to bloodstains, the stain glows in the dark, and even if it's in small trace amounts. However, luminol use is associated with a number of disadvantages and should only be used as a last resort. So it's really good at finding this hidden blood, but it is presumptive, which means it can actually give some false positives, right? And then you can also have the loss of genetic markers. So for example, this picture of this bathtub, if you suspected that there may be blood here, before you spray the luminol, you would probably take several swabs of the area that you think would have blood first, and then spray it with luminol and snap the pictures and make sure you keep that as part of the record, the documentation. So here's that picture I was talking about. If you take a look here on the left, this is normal. And you would look at this and say, oh, I don't see any blood, and you could look really close and wouldn't really be able to tell anything. But if you flip off the lights and spray luminol, all of a sudden it's glowing. Now, when you do this, when you spray it with luminol, it doesn't glow forever. It's not gonna keep glowing. It actually only glows for a few seconds. So you got to kind of spray and take the pictures while it's glowing. If you wait too long, it'll start to dilute. The other thing is luminol, when we use it, um, has a very short shelf life. It usually comes in a powder, and then you mix it and turn it into a liquid, put it into a spray bottle, and you can spray it on stuff. But once you've mixed it into its liquid form, the shelf life's only 2 or 3 days at best, and then you got to get rid of it. So you don't want to mix it up ahead of time. You're usually going to mix it right there at the scene, and then use it right away. So how is blood collected at a crime scene? All right, so now we're going to get into the collection and packaging a little bit. As biological samples are collected at the scene, it's important to remember the facts you're trying to establish within the investigation. The evidence collected must provide the most information. With blood stains, it's important to collect representative samples from areas away from the action, or the blood stains and spatters that appear different from the majority of the other stains, since these may provide the most information. For example, if you have the scene, a large scene where you have a body and there's lots of blood in the middle of the floor, well, you're going to sample that, obviously, but if you have smaller spatter patterns away from the scene, drips away from the scene, a smear maybe on a door handle away from the scene, you're going to have to take those samples as well. Don't just assume that all the blood belongs to the victim. Blood stains leading away from the scene may have come from an injured suspect or an assailant and may provide additional information leading to their identification. Analysis of biological evidence requires the comparison of evidence with standard reference samples obtained from the victim and suspect. So if we get, for example, if we get let's say we get a suspect and they have some blood that is on their clothing, and we're able to get a sample of the blood from their clothing, we need to match it up with the blood from the victim and see if it matches. It's a comparison method, okay? That's what genetics is. We look at DNA and we compare the two, okay? But it could also be vice versa. If there's blood stain maybe on the victim's clothing and it doesn't match the victim, and then obviously we have a suspect, then we'd have to be able to match it to a sample from the suspect. So you have to consider blood groupings, right? A comparison will be made between the genetic markers present in the victim's blood and the suspect's blood. For example, ABO blood typing found on a suspect's clothing may be carried out for some comparison with a control blood sample taken from the victim. However, unless a control sample has been obtained from the suspect, the ABO blood grouping results wouldn't provide much information and just tell us the blood type. All right, so let's talk about how we collect this stuff. There's two types of stains you're going to be coming into contact with. You could have dry blood stains or wet stains. If it's a dry stain, then if possible, try to transport the entire item to the laboratory. Now that's not always possible, but if it's a blood stain that's on a shirt and it's dry, you're going to take the entire shirt and send it off to the lab for analysis. You would package it in a paper bag. Always use paper whenever possible for blood, and I'll give you the one time where you can use plastic for a short period of time on the next page or next slide. If the item's extremely large or not easily transported, then samples can be removed from the item. For example, if it's on a large rug and you can't take the rug, it's in there, right? But if the staining's on the rug, you would make a cut out of the rug. You would cut a section of the rug out and send that section in. You can, if it's on a countertop and it's a dry stain, you can scrape off basically dried scrapings into a bindle and send that in. Or you can lift it off with a cotton swab, wet in a cotton swab with some distilled water, and so on. If they're wet stains are collected by absorbing the stain onto long cotton threads or small squares of gauze, cotton gauze, or a cotton tip swab, right? And if you come back here, this would be what they're talking about. It's basically almost like a Q-tip, but they're a little bit longer. And then you put them into something called a swab box. You can see this right over here. Sorry, so this is the cotton at the end, and then this is the box or the container that you would put it in. to collect it. Okay. However, if you're collecting the entire item, let's say it's a shirt again, but this time it's dripping with blood. It's just completely soaked. So the general rule is no packaging in plastic. And again, this goes back to your research that you're going to do. If you package it in plastic and seal it and keep it in there for a long period of time, it actually leads to the degradation of the DNA, right? You could have bacteria in there, you could have different things in there that basically cause the DNA, the useful DNA in that blood to kind of get degraded or contaminated, right? So as a general rule, no packaging in plastic. However, it may be done as long as it is packaged in this container for less than 2 hours. This is a temporary situation. So if it's dripping with blood, you obviously aren't going to put it into a paper container, a paper bag, or envelope, or something like that, because it's going to soak through and it's going to get all over the place, and you don't want to do that. So you could put it into a plastic bag for a temporary solution until you get it to the lab. And once the item arrives at the lab, you would take that item and put it into one of these large drying cabinets and dry it out. Now, be careful with your method you use for drying as well, because again, heat can degrade DNA, so you wouldn't use like a hair dryer or something. So you have to drip dry or use these drying cabinets, and remember, high heat can destroy the DNA. Once that's done, you would repackage it along with the envelope, and then you would have to store it, right? How do you preserve this? Because you might not be ready to analyze it right away, because it does you no good to analyze it unless you have something to compare it to. So you might not analyze it right away, and then later on, as the investigation continues, all of a sudden now we have a suspect and we have a sample from the suspect, and now we can do a comparison. And that's when we would pull it out of our storage. So blood and other biological evidence should be refrigerated whenever possible to lessen the possible degradation of the evidence during storage. So you would keep it in a refrigerator unit, and they have various types. Obviously, they have bigger ones and so on. But you would keep it in there until you're ready to do any kind of analysis. So that's it for this week. That takes care of the notes. Later at the end of the week, we'll probably I will open up the test for this unit, and we'll get that done. And I will be talking to you a little bit more about the check for understanding, and if you have questions about the writing assignment, the 500-word essay that you're going to do on the DNA, I will talk to you about that later this week as well. All right, guys, have a great day.